This is Bio 182, Lecture 1.1, Foundations. One of the first questions you probably thought when you enrolled in this course is, what will we cover in Bio 182? There is a list of course competencies. Those are in the notes for this slide as well as on your syllabus. So if you are interested in the specific course competencies, those are the two places that you can find them. However, in general, we're going to be discussing organismal biology more than genetics and cellular biology. We're going to get to talk about the ecology and evolution of organisms instead of their genetics and cellular processes. Because we're going to be talking more about life and living things, we need to define what we mean by life. Biology is itself the scientific study of life. The scope of biology stretches across an enormous diversity of life on Earth, from bacteria to fungi to plants and animals. Any living thing on Earth all share certain characteristics that define them as life. Some of those characteristics are that they have order. They are made of cells. Those cells have machinery. They have genetics that create all of those things. They have some type of regulation where the cells and individuals will regulate their temperature or the amount of water or salt that they contain. They remove waste. Growth and development, where all organisms on Earth have the ability to grow and develop throughout their lifetimes. All living things use energy. You would have spent quite a bit of time on this in Bio 181 when you're talking about cellular respiration and photosynthesis. All living things respond to their environment. If they are stimulated through various ways, plants and animals both will respond to stimuli in their environment. All living things have the ability to reproduce and have heredity through their genetics. And all living things are subject to the processes of evolution, which we will go into quite a bit of detail on throughout this semester. When we're talking about different types of life, there are different levels of biological organization. Life is structured on a size scale ranging from molecular to global. When we're talking about biological organization, we start with the smallest components of life, which would be organelles. Multiple organelles make up cells, multiple cells make up tissues, multiple tissues make up organs and organ systems. Those are the levels of biological organization that you would have focused on in Bio 181. In Bio 182, we're going to focus more on organisms as a whole, populations of organisms, communities of organisms, ecosystems, and the biosphere as a whole. Biology is, by its nature, a hypothesis-driven science. A hypothesis-driven science means that it is a formal process of inquiry, and the scientific method consists of a series of steps. The key element of the scientific method is that it is a hypothesis-driven science. What this means is that a hypothesis is a proposed explanation for a set of observations, and that hypothesis is an idea on trial. That hypothesis is then tested by performing an experiment to see whether results are as we predicted. This deductive reasoning takes the form of that if-then logic. If this occurs, then I expect this to happen. If this is true, then this should occur. Those are those hypotheses that we're talking about. The scientific method is used to study all parts of science. In general, when we're talking about the scientific method, we assume that it has this linear process where we have an observation that is made. That observation sparks interesting questions. We formulate hypotheses regarding those observations. We develop a test that will allow us to test the predictions, and then we implement that test and gather data to test our predictions. From that, we are then able to explain the results of our experiment and decide whether or not our hypothesis was supported or not. In reality, science is a continuing process. We are constantly refining our hypotheses, making new observations, doing new experiments that lead to more and interesting questions, and the scope of knowledge that we have is constantly expanding. One good example of this scientific method at work in our current environment is the use of masks due to the COVID-19 pandemic. At the beginning, the CDC had said that masks were not necessary because we thought that COVID-19 was being spread through the same mechanisms as the common cold. As we were able to do more research into COVID-19 mechanisms, we have realized that it is spread through droplets in the air, and therefore masks are useful in preventing the spread of COVID-19. We've also had several groups of scientists doing experiments on the types of masks that are effective and which ones are most effective. And so that data is constantly evolving. One complaint from the public is often that scientists are constantly changing their minds. And in reality, scientists are doing their jobs by testing the hypotheses of others. They're testing their own hypotheses, creating new hypotheses based on research, doing similar experiments but in different ways, and measuring things in different ways. Science should always be progressing forward, and that is what it is doing in this context. When we're talking about hypothesis-driven science, a null hypothesis is the expectation that there are no differences between two variables. So a null hypothesis is essentially what you expect to happen if there is no effect of your variables. If the variables have no difference, then we expect the null hypothesis to be true. An alternate hypothesis is the expectation that there are differences between two variables, that the variables are going to cause a different outcome. So the null hypothesis is what we expect if there are no differences between the variables. The alternate hypothesis is what we expect to happen if there is a difference based on the variables. So we have this example of cats, and we are wondering if cats show a preference for food based on shape. So the null hypothesis is that cats do not have a preference for food based on the shape of the food. The alternative hypothesis is that the cats do have a preference for a certain shape. For example, if a cat has been fed rectangle-shaped food its entire life, it will continue to prefer that when it's presented that option with another. So the decision to reject a hypothesis is based on statistical analysis of data. Statistics are very important for any science and for any study. Statistics are what we base our conclusions on, whether or not there is strong enough evidence to reject our null hypothesis and accept our alternative hypothesis, or if that statistical data is not strong enough to reject the null hypothesis. What we use to determine that is called a p-value. Generally, if a p-value is less than 0.05, it is considered strong evidence that we can reject the null hypothesis. So when you're reading scientific studies and you're learning more about, or when you're reading a scientific study and you are reading about the statistics, a strong p-value should be lower than 0.05. If it's above that, then we generally consider that to not be reason enough to reject the null hypothesis. And hopefully you guys will have an opportunity to take some statistics courses during your college degree to get a lot more clarification on the importance and use of statistics in science. So here's an example of how we can accept or reject null hypotheses. So one of these two snakes is a coral snake. Based on their appearance, which one would you want to pick up? One of them will kill you and the other one is harmless. So in this example, one of the snakes is mimicking the other in order to trick possible predators into not wanting to eat it. They're essentially pretending to be poisonous and dangerous when they're actually a completely benign organism. And we're going to look a little bit more in depth at a study done by Brody in 1993 on coral snakes. So when we're talking about aposematic coloration, what we mean is that it's a warning to others, including predators, that the signaler has chemical or physical defenses. So up in the top right, we have a monarch butterfly. Monarch butterflies are toxic if they are eaten by any kind of predator. Monarch butterflies eat milkweed. Milkweed itself is toxic. The monarch butterflies consolidate those toxins into their tissues, which then makes them toxic. And so the bright color on the butterfly says to a predator, I am bright, I am scary, and I probably don't taste very good. I will make you sick, don't eat me. Similar situation is going on with poison dart frogs. They have a secretion that comes out of their skin that is toxic, and they are bright colored as a warning to predators that says, don't eat me, I will make you incredibly sick. At the bottom of the slide, there's several insects that have the same sort of red and black color pattern, and they're all saying the same thing. I don't taste good, you don't want to eat me, run away. And so what's interesting about these is we usually think of things that could be prey as trying to hide, right? You think of something like a bunny rabbit, small birds, things like that. They're trying to blend in. Animals that have aposematic coloration are able to be bright and colorful because they have a chemical or physical defense that makes them toxic or harmful to a potential predator. So they don't have to hide. They get to look scary and say in bright colors, stay away. You don't want to eat me. Cryptic coloration is the opposite side of the spectrum. So this would be more like camouflage. It's a way to avoid being detected by others. So up in the top right, we have a lizard who blends in very well to the rocks in his habitat. We have a small bird that blends in well to its to the leaves in its environment, and then we have a moth that looks very similar to the leaves on the ground in the environment in which it lives. Usually we think of camouflage as being something that is dull in color and makes it difficult to see the animal. But in the case of this bird, or even if you think about a tiger, A tiger is hiding in an attempt to trick its prey so that they don't see it. Bright colors sometimes help that individual blend into the environment. In the case of a tiger, the stripes on it make it look as though it blends into the grass that it's stalking its prey through, and it's very difficult for the prey animals to determine where the tiger is among the grass because it just sort of all looks like shadows together. So cryptic coloration is essentially camouflage, and it's the opposite end of the spectrum from aposematic coloration, which is bright colors that say, don't eat me, I don't taste good. Camouflage is cryptic coloration, and that is a way for a prey item to hide and avoid detection instead of using chemicals or physical defenses to keep predators from trying to consume them. So going back to our coral snakes, Brodie created a hypothesis that it was the tricolor of the coral snakes that made birds not want to attack them. And so he made it so he made pretend snakes out of modeling clay of different colors, and whether they were brown in color or tricolored, and then put them on either natural backgrounds or an artificial background. So the null hypothesis would be that birds attack all snakes equally on all surfaces, which would mean that the coloration is not going to affect how frequently a bird attacks a snake. The first alternate hypothesis is that birds actively avoid tricolor snakes, which would be an aposematic coloration hypothesis, that tricolor on the coral snakes is going to make them dangerous and poisonous, and therefore predators should leave them alone. The second alternate hypothesis is that birds don't notice tricolor snakes because they are camouflaged. And so with the fake snakes that he made out of modeling clay, he would then be able to count the number of peck marks in each of them to determine how many birds had attacked each of the snakes, whether they were brown and blended into their environment or tricolor and looked more like that aposematic coral snake. So when we make a hypothesis, we need to know what it would look like if the null hypothesis were true or if either of the alternative hypotheses were true. So the first is the null hypothesis, which is that the rate of attack for all colors on all surfaces is the same. So whether the snake is tricolored or natural, and whether it's on a natural or a white background, they should receive the same number of bird attacks. The aposematic coloration hypothesis is that on natural backgrounds, tricolor snakes will be attacked less than brown snakes. So on a natural background, also on a white background, tricolor snakes will be attacked less than brown snakes. So in this case, we're saying that the snakes are aposematic, they are dangerous, their predators know that they are dangerous, and therefore they're going to avoid them regardless of the background that they are on. The next is camouflage coloration. And so in this hypothesis, we're going to say that on natural backgrounds, tricolor snakes will be attacked less than brown snakes because for some reason their coloration is allowing them to be camouflaged on that natural background. Or on a white background, both colors of snake will be attacked at the same frequency because on a white background, they are no longer camouflaged. What Brodie found was actually support for the aposematic coloration. Tricolored snakes were attacked less on both natural and white background, and therefore we know that coral snake color patterns are aposematic and do not provide camouflage. So this is just an example of how we use the scientific method to make an observation, make hypotheses about that, or create a question, create hypotheses, design an experiment, look at the results, and then create conclusions based on those results. In science, we have theories, and when many experiments support a common hypothesis, it causes that to become a theory. It only takes one experiment to reject a hypothesis, but it takes a lot of experiments to reject a theory. So you never are able to prove a theory, you're never able to prove a theory, you only are able to find support for it. So in this case, it's like a table with multiple legs. It's not just supported by 2 or 3 legs of support. It is supported by many, many, many studies that support the same hypothesis that allows it to be a theory. Unlike other sciences, there are no laws in biology. We only have theories in biology. So there are several foundations to modern biology, and most of them we would consider theories. We're going to go through these briefly. The first is cell theory. So this is the theory that cells are the basic structural units of all living things. Cells are distinct entities, but are also the building blocks for complex organisms. And all cells come from pre-existing cells. This I believe you should have covered in Bio 181, but this is the basic units of cell theory, as well as all cells are similar in chemical composition. And most of the chemical reactions of life occur within cells. Evolutionary theory, which we will spend quite a bit of time studying in Bio 182, explains the origin of life and the diversity of life, how we have so many different species and so much variety of life on planet Earth is through the process of evolution, through natural selection. Evolution should be mechanistic and measurable or quantitative. We should be able to see evolution happening. It happens very slowly, but it does happen through time. And it is one of the foundations for modern science. Gene theory or omics. Again, this one you should have spent quite a bit of time talking about in Bio 181. So gene theory is that DNA or deoxyribonucleic acid molecules are made of nucleotide subunits. Triplets of nucleotides code for amino acids, and these amino acids are the building blocks of proteins. Those proteins and that DNA are life's blueprint for all living organisms, and they all use the same code, the universal triplet code of DNA. If this is a little hazy, I would suggest going back and reviewing this a little bit. It will help when we talk more about evolution. Homeostasis. So living organisms regulate their internal environment. This can be different ways. Living organisms can regulate their temperature, their salt balance, their nutrients, and waste materials. And it's not only animal cells that do this, plant cells do this as well. So when we're talking about homeostasis, in this process, there's going to be a stimulus, some type of receptor response to that stimulus, such as nerves in your skin, glands in your body, hormones, things like that. There's an integrator, such as your brain, and then an effector, such as a muscle or a gland, and it will respond. And there's a constant feedback loop that says, so if we're talking about temperature, your receptor would be your nerve cells. They sense that the temperature is hot. Therefore, your body starts to sweat in an attempt to cool you down. And until your body re-regulates, your body will continue to react in that way. Once your body has cooled down, you will stop sweating. So that's sort of that feedback loop that we're talking about. The last one that we're going to talk about is behavior. Behavior is my absolute favorite thing to talk about. This is what my PhD is in. So organisms interact with each other and the environment. And conflict and cooperation have important consequences for living organisms. We don't usually think of plants as behaving, but plants actually do sort of have behaviors. And those behaviors and conflict and cooperation occur through the process of evolution. It is all intertwined. And we will get to talk a little bit more in depth about animal behavior, but these are sort of some of the things that we're going to focus on for this semester. That is all that we have for this slide, or for this set of slides, and we will move on.